member of the academic staff within the Department of Anatomy, University of Otago, and part of the leadership team of our National Dementia Prevention Research Clinics. These aim to understand neurodegeneration from individual, cultural and biological perspectives, including biomarkers of disease risk and progression. Initially, I was fascinated by the power of emerging molecular techniques and how these could be used to understand complex biology or diseases. This led to the current focus of our work, which is to develop an easy-to-use blood biomarker for Alzheimer's or Parkinson's disease. Alongside monitoring changes in microRNA patterns in blood plasma as early markers of Alzheimer's or Parkinson's disease, we study a protein which is called SAPA as a potential agent, aiming to understand its mode of action at synapses and how it strengthens connections within the brain. We use custom-designed TaqMan Array Cards to measure the levels of microRNA in plasma and other biofluids. We have chosen this platform as we have found it to be highly reproducible, highly sensitive, and straightforward to perform. RNA is isolated from biofluids using MicroRNA Paris RNA isolation kit, and then converted to complementary DNA using applied Biosystems TaqMan microRNA reverse transcription kit. Following preamplification, 14 cycles, microRNA profiles are generated using custom-designed TaqMan low-density arrays. We initially screened more than 700 microRNA, but have narrowed the list down to a group of 48. This means we can process 8 samples simultaneously, producing a large amount of data. We've worked hard on streamlining the QC data process and fine-tuning the data analysis to our needs. We use these tools because the process is really straightforward. This platform enables the simultaneous quantification of 8 samples per card, therefore reducing potential inter-subject variability. From assessment of technical replicates and interplate variability, we have shown that this platform yields highly reproducible results. Assay variability is less than 10%. This design also allows for assessment of 48 microRNA per participant. Thermo Fisher Scientific has been instrumental in our research from the very beginning. We are very grateful for that.
We use a combination of TaqMan gene expression master mix, as well as a custom TaqMan array cards for microRNA analysis. The combination of these tools has proven to be quite robust for our screening process. We also use individual assays to assess expression of specific microRNA of interest. While using our current methods, these tools allow us to screen microRNA expression in 8 samples simultaneously. This has allowed us to screen hundreds of biofluid samples. Our Alzheimer's work is published, and we have intellectual property protection underway. Many of our postgrad students have used this technology to support their own thesis work. Well, first, the ability to screen multiple microRNAs simultaneously, as well as in multiple biofluid samples. Second, the strong reproducibility of the data. First, the array data are analyzed in the software environment R via the high-throughput qPCR package. Once all the quality control assessment is completed, we then normalize the data using the normagene algorithm. Then we can proceed to find which microRNA are significantly expressed.
Yes, we were pleased to be able to contact the tech support team who were able to share with us updated protocols to our systems. We are currently pursuing the use of this technology to measure microRNA levels in tear fluids and blood spots, as we see this as a way of being able to include a wider representation of society within our biomarker studies.